Hello, my name is Megan Lewis, and I'm a master's student in biomedical engineering here at the University of Utah. I work in Dr. Bruce Edgar's lab at the Huntsman Cancer Institute, and today I will be presenting our project on the translational regulation of cell cycle genes during contact inhibition using ribosome profiling on human epithelial cells. Contact inhibition is the process by which cells stop proliferation and migration in response to contact with other cells. The loss of contact inhibition is considered to be a hallmark of cancer. Ordinarily, when a tissue is forming or a wound is closing, cells will populate the appropriate area until an adequate cell density is reached. When this happens, signaling instructs them to halt this reproduction. Without the signaling, cells will continue to proliferate in an uncontrolled fashion. Cells may also migrate away from the area, leading to metastases. One of the key points of cell cycle regulation is at the G1 to S phase transition. Signaling from the E2F transcription factors acts through cyclin E and cyclin-dependent kinase 2 to drive expression of many genes needed for DNA synthesis and cell replication. E2Fs are repressed by the retinoblastoma protein, PRB, and become active when cyclin D and CDKs 4 and 6 repress this retinoblastoma protein. The regulation of cyclins, CDKs, and CDK inhibitors is largely considered to be at the level of transcription, meaning the amount of protein is directly related to the amount of mRNA produced. However, there have been several studies demonstrating that the cell cycle can continue to progress even when this model is disturbed. Therefore, there may be regulatory elements missing. Our lab proposes that a translationally regulated, growth-dependent protein may be the missing piece. To identify this protein or proteins, we are analyzing contact inhibition in healthy human retinal pigmented epithelial cells using ribosome profiling. The fluorescence image to the right displays a semi-confluent culture of these RPE1 cells with actin filaments stained in red and nuclei stained in blue. Ribosome profiling allows us to identify proteins that are translationally regulated during contact inhibition. It is important to identify the time points and densities at which contact inhibition occurs, so we constructed a growth curve of the RPE cells from multiple cultures plated at the same starting density. We then collected cells at 20-hour intervals from 40 to 120 hours. Collected cells were flash frozen and then stored at negative 80 degrees Celsius until all the samples were collected. Then the cells were lysed in RNA-free conditions with cyclohexamide to halt the ribosomes. This allowed us to purify the RNA from the cells. Lysates were then treated with RNase to digest any RNA that was not protected by a ribosome. This means that any RNA preserved was a transcript that was actively being translated at the moment the cells were flash frozen. Next, the ribosomes were isolated by ultracentrifugation through a sucrose cushion. The ribosomes are then removed to reveal the ribosome-protected fragments of mRNA, also known as RPFs. The RPFs are then cloned into DNA libraries with unique identifier sequences added. The libraries are then deep sequenced by Illumina sequencing. We are currently awaiting the results of the sequencing on our samples. The sequence fragments are then aligned to a reference human genome. This allows the sequences to be associated with their genes and the transcripts per gene to be quantified. Differential expression analysis with DE-seq2 will then reveal which genes are translationally regulated. For a simple example, if there are two transcripts made for one gene and only one RPF, that gene is translationally downregulated. If there is the same amount of translation as transcription, then that gene is not translationally regulated. Lastly, if translational activity is higher than the transcriptional, the gene is translationally upregulated. Now, realistically, transcripts will have many ribosomes translating them simultaneously, and it is a polysome. The ratios I chose here were just to be simple examples. Identifying and understanding the genes that are regulating the cell cycle during contact inhibition in healthy cells will shed light on the missing regulation in cancer cells. Restoring this regulation could be a big step in halting cancer. And with that, I would like to thank my advisor, Bruce Edgar, and Stephen Pronovo, my mentor, and to Matthew and Rachel for putting this event together. Thank you.